Good afternoon, everyone. Can you hear me okay? All right. Did everybody have a nice weekend? Okay, good. Okay, so today the topic of our next 2 hours is gluconeogenesis. So this is kind of rounding out rounding out our discussion of fasted state metabolism that we started on Friday with glycogen metabolism. So as usual, we'll focus on the main function of the pathway, the key substrates, the key products, and the regulatory steps. There's a lot of overlap with glycolysis, fortunately, so there's a lot of stuff that we're going to just skip over or not spend a lot of time with. In terms of our pathologies, there's one, the glucose 6-phosphatase deficiency, von Gierke's disease. We're going to revisit that because that has consequences for gluconeogenesis as well. Okay. Are there questions before we get started? And this is just a outline of where we're going. As far as those clinical cases at the end, these are the two cases. This first one, this is quite a sad case for just trigger warning in case anyone is upset by that sort of stuff. The bottom one is a much nicer case. I'm going to try something a little bit different. I've got the case information for both of these time released, so rather than having to read everything off the screen here, it should show up in your Canvas slot at like 2 o'clock. Okay. Okay. Shall we start with a little clicker quiz? Okay, should we take a look here? Okay, looks like just a bare majority of people selected the correct answer, amylopectin. So amylose and amylopectin are the two main dietary starches. Amylose is an unbranched chain of glucose molecules, and amylopectin is a branched chain, or a branched network. And that's shown here. So amylopectin has the 1,4 linkages between glucose molecules to make the linear portions, and then the branch points are made by 1,6 linkages. Okay, very similar to glycogen. Okay, so this is definitely one of those big concepts that you'll want to keep front and center with the glycogen storage diseases. Pretty easy to understand though, right? Okay, good. Okay, so on to our current topic. Ooh, the gold rush. I suppose a lot of people starved during that because there were people who were kind of given false promises and false hope of riches, and then sent out into what was then an untamed wilderness. The answer is actually the Minnesota experiments. Yep. Has anybody heard of Ansel Keys? He was the leader of that. So this was a series of experiments. Let's see, were any of you alive to see Memorial Stadium on campus, or was that demolished? I think that was demolished in the late 90s. So Memorial Stadium was where the Gophers used to play football. During World War II, conscientious objectors primarily were signed up for an experiment in which they were essentially starved to death. They were on I'm sorry, I don't think anyone died. They were put on a starvation diet, and various physiology and psychology and et cetera parameters were measured in these people for several months until they were near death, and then the refeeding physiology was also studied. So these were published in a couple of books about 70 some years ago, and these remain today seminal works on the physiology of human starvation. Okay, so a lot of what we know, a lot of what we'll be presenting over the next couple of hours has come from work that was done by our institution here at the University of Minnesota. So, kind of interesting. Okay. Okay, what do you think here? Okay, I think ketone bodies is maybe a good guess based on what we've talked about, but really the best answer is this one right here, glucose. Okay, did one person answer that? It's like 3% of the class. So it's ketone body production is a normal physiological response to calorie deprivation or starvation. However, the brain is going to prefer to use glucose until you get into much more advanced starvation than this. Okay, so what are we at? 9 days after his last meal. Here he would be brain would be decreasing glucose use and increasing ketone body utilization, but glucose would still be the major source of the brain calories at this time. Okay. Okay. I'll just go over these, and we'll talk about these at the end of the course or end of the lecture. Okay, so if we consider the abundance of different substrates within our body after we have eaten our last meal at time 0, and then we go for a month or more without eating. So you'll notice, I guess, first, I think the kind of remarkable thing really is that the blood glucose concentrations drop, but you can go over a month without eating and your blood glucose concentration doesn't drop below 70 milligrams per deciliter or so. So that's kind of remarkable, and really the body is modifying its metabolism so that it can sustain that process, because we know that there are certain tissues that don't have any metabolic flexibility. They can only use glucose as a fuel. All right. Okay, so as the glucose drops, the concentrations of free fatty acids will increase. So there's hormone-sensitive lipase. This is the lipase in fat tissue that clips off individual fatty acids from triacylglycerol. So the hormone-sensitive lipase, remember, that is inhibited by insulin and then activated by glucagon and the catecholamines. So as those endocrine signals are increasing, you'll get increased free fatty acids that are released from fat stores, which are our biggest source of stored energy in the body. Those free fatty acids then, those are used to provide energy to the liver by way of fatty acid beta oxidation. They're also providing energy for skeletal muscle and organ tissue to gain ATP through fatty acid beta oxidation. Now, unlike the muscle that's using the beta oxidation to generate energy, and then the acetyl-CoA product is further oxidized down to carbon dioxide in the tricarboxylic acid cycle. In the liver, the beta oxidation of fatty acids is making energy, but the acetyl-CoA that's generated from fatty acid beta oxidation within the liver is going to be devoted to the synthesis of ketone bodies, acetoacetate, and then the more abundant beta hydroxybutyrate. So the ketone bodies then, as the availability of carbohydrate energy is decreased, ketone bodies become an alternate fuel that can be used by skeletal muscle, organs, and ultimately the brain can use ketone bodies in very advanced starvation. Okay, so if we just consider the carbohydrates and look at this glucose a little bit more in starvation, immediately after a meal, the glucose that's being consumed or being oxidized, that glucose is coming from the carbohydrates in that last meal. Okay, so that's indicated by the green here. And you can see that in that period immediately after the meal, that we're using a lot more glucose than when we're in the fasted or starved state. Okay, so in this fed state metabolism, or the postprandial period, it's sometimes called, glucose is our preferred fuel, and that glucose is coming from our last meal. Okay, now when that is exhausted, then we're getting, now remember, the glucose concentration in our blood doesn't decrease, right? Or doesn't decrease very much, but we don't have any more of the the dietary carbohydrates. So in the fasted state and the starved state, the glucose in our blood is coming from hepatic glucose output or from liver sources of glucose. We talked about glycogen metabolism and the role of glycogenolysis in the fasted state on Friday. So the first source of hepatic glucose is going to be from the glycogen stores in the liver. As the glycogen stores are being depleted, then gluconeogenesis ramps up. And the products of these two processes are the same. They're both making glucose that's exported out into the blood, but the sources of those glucose are different. So in glycogenolysis, the glucose is coming from stuff that's already in the liver, glycogen, whereas in gluconeogenesis, the substrates for gluconeogenesis are coming from outside of the liver. And there are some diverse processes that can generate those substrates, as we'll elaborate on shortly. Let's see. Now, you'll notice here that the gluconeogenesis increase ramps up, and then if you don't have, say, if you eat dinner at 6 o'clock and you normally eat breakfast at 6 o'clock in the morning, and then you don't do that, say, the gluconeogenesis rate is going to drop off from its maximal overnight rate. And this is because gluconeogenesis using protein-derived substrates, using amino acids as substrates, that's not sustainable for the long term. So the reason that we decrease our gluconeogenesis rate here is because we want to sustain organ protein and muscle protein, because that's going to allow us to find our next meal, essentially. Okay. All right. So if we look at, I have this table that's kind of a summary of the different states. So I think the Homer question, that would have been in like intermediate starvation. Okay, so here we've got hepatic gluconeogenesis. There are also some cells in the kidney that can do gluconeogenesis, and I'm just going to leave it at that and not say anything more about it. So just for the purposes of this discussion, just consider gluconeogenesis to be a liver process. Okay, so the brain then, the brain is going to be using glucose as its preferred fuel up until we get to very advanced starvation. Okay, and when we're, in a month or so of no food, then the brain is going to be using ketone bodies as its major fuel. So the tissues using glucose here, red blood cells, these are the ones that are really, that's the rationale for why we have this gluconeogenesis happening, because red blood cells are committed to glucose metabolism as their only source of ATP. In terms of the endocrine response, so the insulin, of course, is the major hormone that's mediating carbohydrate uptake, utilization, and storage in the fed state. Then we get glucagon as the major fasted hormone, and as we increase our stress, then we'll get cortisol, and the catecholamine hormones will increase, and they'll contribute to that fasted and starved state metabolism. Okay, so gluconeogenesis, instead of glycolysis going from north to south, gluconeogenesis is following the same pathway on our metabolic map, but we're going from generally south to north here. Okay, so the purpose of gluconeogenesis is to use lactate and glycerol and amino acids as substrates to build glucose and to get it out into the blood to sustain those cells that are absolutely dependent on it. There are a lot of overlapping enzymes with glycolysis. Okay, so there are 3 important reactions that are not catalyzed by glycolytic enzymes, but all of the rest of the enzymes that are responsible for converting pyruvate A into glucose, those are all glycolytic enzymes. Okay, so another thing to mention here is that there's a lot of overlap with glycolysis. Okay, so I think we've seen this cartoon before summarizing the carbohydrate metabolism in the fed, fasted, and starved states. So after just a typical overnight fast, you're going to be using the glycogen as your major source of hepatic glucose output. And then as glycogen is depleted, then you're going to shift to gluconeogenesis, which is using external or extra hepatic substrates, if you will, to make glucose. In the starved state, you have no more glycogen left, so then you're essentially reliant completely on gluconeogenesis for sustaining blood glucose levels. Now, the major, in say normal day-to-day eating and fasting cycles, the major source that's going to sustain gluconeogenesis after like an overnight fast is muscle breakdown and liberating amino acids from muscle that can be used to turn into glucose here. Okay. That is assuming, or your body is assuming, that you're going to have breakfast and you're going to switch back into fed state metabolism and you won't have to continue breaking down your muscle protein. Okay? So if you consider the different substrates that can contribute to, or that can be gluconeogenic, proteolysis, or breaking down muscle and organ protein to provide amino acids for the gluconeogenesis. As I mentioned earlier, that's not sustainable long term. Okay, so in starved metabolism, we're decreasing this and increasing fat metabolism because we can't just continue using proteolysis to sustain gluconeogenesis for a month without calories, because if we did that, we wouldn't have any muscle to do any work at all. we die. Okay, is that clear? So maybe put a little down arrow on this proteolysis for long-term starvation. But just after an overnight fast, we do a lot of protein breakdown. So there's this kind of daily cycle of breaking down protein in the fasted state and then building up protein in the fed state. Okay, so let's consider our different gluconeogenic substrates that we can derive from lipolysis, proteolysis, and fermentation. Fermentation, whoops. Yeah, fermentation down here, that's another name for anaerobic glycolysis. I think you've heard that before. Okay, so anaerobic glycolysis, we know that the general products of glycolysis are pyruvate, but in the absence of oxygen, we will convert that pyruvate into the alcohol lactate, right? So lactate then, if a red blood cell is using glucose, it's going to be generating lactate. That can go out into the blood. The liver then can take up lactate through the monocarboxylate transporter and then oxidize it to pyruvate. So pyruvate is a key gluconeogenic intermediate when we're using lactate as a substrate. Also, many amino acids are gluconeogenic or can be gluconeogenic. And many of them are converted into alanine, and then alanine can be transaminated into pyruvate. Others are made into pyruvate without going into alanine, such as serine, for example. But just generally, alanine is one of the key gluconeogenic amino acids. So when we have muscle protein degradation and anaerobic glycolysis, they all converge on pyruvate as a common gluconeogenic intermediate. Okay. That's distinct from the substrates that can be gained by lipolysis or breakdown of triacylglycerol. Triacylglycerol, the structure of triacylglycerol, I'm not sure if we've seen that before. It's the glycerol alcohol backbone, and then there's a fatty acid like palmitate on each one of these carbons. Okay? So when hormone-sensitive lipase is active, if we're using one molecule of triacylglycerol, hormone-sensitive lipase will make 3 free fatty acids and 1 molecule of glycerol. So glycerol, this can be converted into glycerol 3-phosphate by glycerol kinase, and this glycerol kinase is only expressed in the liver, and that has some consequences for who can store fat when that we'll get to in a moment. The glycerol 3-phosphate then, this can be interconverted with the triose phosphate steps, the dihydroxyacetone phosphate, and then glyceraldehyde 3-phosphate. So this, when we're using glycerol as a substrate for gluconeogenesis, we can go into the gluconeogenic pathway further north than pyruvate, if you will. Okay, so I think that'll be clear towards the end of the hour here. Okay, if we can just review glycolysis. Now, the key regulatory steps in glycolysis are, of course, the phosphofructokinase 1, right? This one, in here, phosphofructokinase 1 has the allosteric regulation by AMP and inhibited by ATP and activated by the fructose 2,6-bisphosphate, and I think I might be forgetting one. Okay, anyways, we've got allosteric regulation at this step. The glucokinase and hexokinase, the different Michaelis-Menten constants or their differing affinities for glucose, that was a key regulator at this step. And then at the pyruvate kinase step, we had regulation with different isoforms of the enzyme, and the different isoforms had different regulation by phosphorylation and different allosteric regulation. Does that sound familiar? Okay, so those same steps, the key regulatory steps, here, here, and here. The key regulatory steps in glycolysis are also the key regulatory steps in gluconeogenesis. Okay, so if you consider gluconeogenesis as just being the reverse of glycolysis, it's using all the same enzymes except for the enzymes that are catalyzing those three key regulatory steps. So we've got glucokinase / hexokinase, first phosphofructokinase 1, and then pyruvate kinase. These regulatory steps are catalyzed by different enzyme complexes in gluconeogenesis, but some of the regulation is coordinated. Okay, so let's look at that in a little more detail. The glucokinase step, that's catalyzed by glucose 6-phosphatase in gluconeogenesis. The phosphofructokinase 1, the PFK1 reaction, that is catalyzed by fructose 1,6-bisphosphatase. The pyruvate kinase step, this is catalyzed, so if we're going from phosphoenolpyruvate through pyruvate kinase to pyruvate in glycolysis, the reverse reaction is a multi-step process that has to dip down into the TCA cycle, and then out of the TCA cycle and into the mitochondria. And anyways, it's a multi-step process, but the key regulatory step is the phosphoenolpyruvate carboxykinase. So the major regulation for all of these is transcriptional regulation, both by cortisol and by protein kinase A dependent transcription factors. We also have the fructose 1,6-bisphosphatase. This is has a coordinate allosteric regulation with PFK1. So PFK1, remember, is activated by the fructose 2,6-bisphosphate. That same little second messenger modulator is acting to inhibit the phosphatase. Okay, so it's turning on glycolysis and turning off gluconeogenesis. Okay, so just looking at glycolysis and gluconeogenesis side by side, everything that's done by glycolysis going from north to south, the same enzymes are doing those same steps in gluconeogenesis going from south to north, with the exception of these three key steps. Is that all good? Okay. What questions do you have at this point? Carry on. Okay, so I will go through in a little bit of detail, starting with the conversion of phosphoenolpyruvate to pyruvate in glycolysis. This is a single-step conversion, substrate level phosphorylation that's taking a phosphate from phosphoenolpyruvate and adding it to ADP. The reverse reaction, and so that's a single step in glycolysis. In gluconeogenesis, it's a multi-step process. Okay, so in gluconeogenesis, we can get pyruvate both from the transamination of alanine, or we can get it from the oxidation of lactate. Okay, so both ways, two gluconeogenic substrates can enter into gluconeogenesis as pyruvate. Now, pyruvate then, of course, has multiple metabolic fates within the cell. We could go through pyruvate dehydrogenase, for example, to make acetyl-CoA. That's where we would go if we were doing the TCA cycle, of course. That's also where we would be going in a condition of excess carbohydrate, where we were wanting to synthesize fatty acids and store some of that excess carbohydrate as fat. We would be going through this direction. Now, in the liver, we're doing gluconeogenesis at the same time we're doing a lot of fatty acid beta oxidation. So the consequence there is that we have a lot of NADH around. NADH then is going to be inhibiting the pyruvate dehydrogenase reaction. Okay, so that's going to, because NADH is a product of the pyruvate dehydrogenase reaction, that's going to block it from going this way, and then if you block that way, it's going to flow through the path of least resistance, and in the gluconeogenesis situation, that enzyme is the pyruvate carboxylase. So that's the biotin-dependent enzyme that is adding a carbon from carbon dioxide to pyruvate to make the 4-carbon oxaloacetate. Oxaloacetate then, this leaves the mitochondria. So just to make this simple, and then I'll make it a little bit more complicated, but okay, the simple part is oxaloacetate in the mitochondria goes to oxaloacetate in the cytoplasm. Okay, and it can go either through malate or aspartate to get out of the mitochondria. So there's no oxaloacetate transporter across the intermitochondrial membrane. Now, you know, I think I'm getting quite a bit ahead of myself here. So maybe I'll pause and see where I'm at with my slides here. Okay, I guess I'm not super ahead of myself. All right, so the pyruvate carboxylase reaction, this is one of those 4 enzymes that's dependent on biotin and performs a carboxylation. It's taking the 3-carbon pyruvate and making a 4-carbon oxaloacetate. We looked at this reaction back when we were talking about the TCA cycle in the context of an anaplerotic reaction, right? So this oxaloacetate, this could be another bucket in our Ferris wheel, right? And take 2 carbons from acetyl-CoA and become citrate and provide more capacity for our TCA cycle. Okay, so that's one potential function of this reaction. In the context of gluconeogenesis, this oxaloacetate has to leave the mitochondria and become a substrate for a phosphoenol pyruvate carboxykinase, or removing a carbon from the oxaloacetate. So in order to get from the oxaloacetate into the mitochondria to the oxaloacetate into the cytoplasm, the oxaloacetate can either be transaminated into a amino acid, aspartate. In this case, the aspartate is leaving the mitochondria through an aspartate glutamate antiporter. So once the aspartate is out of the mitochondria into the cytoplasm, the reverse transamination reaction will occur and regenerate oxaloacetate. Okay, so this is a transamination route of getting the oxaloacetate out of the mitochondria, and it's depending on cytosolic and mitochondrial isoforms of the same transaminase enzyme. Okay, and like most of the transaminases, these are using the alpha-ketoglutarate as a universal amine acceptor and glutamate as a universal amine donor. Okay, so these are kind of always around in the cell, ready to either accept an amine group or to donate an amine group in a transamination reaction. Okay, so the other option, one way to get oxaloacetate out of the mitochondria is to convert it into aspartate. The other way is to convert it into malates. So oxaloacetate, this could be a substrate for malate dehydrogenase. And remember, if we're in a high fatty acid beta oxidation environment, we're having a lot of NADH around, so that's going to promote the malate dehydrogenase running in reverse and essentially and generating malate. Okay, so malate then, this can leave the mitochondria through a different aspartate malate shuttle system, the malate aspartate shuttle, remember that one? Okay. So the malate then can leave the mitochondria and be reconverted into oxaloacetate, again by a cytosolic isoform of the malate dehydrogenase that's in the mitochondria. Okay, so the easiest way to think of this is just oxaloacetate is going from the mitochondria into the cytosol. Which way it goes through, whether it goes through aspartate or whether it goes through malate is going to depend on the relative difference in the reducing power between the mitochondria and the cytosol. Okay, so if you've got enough NADH here that you can push it to malate and then push it back from malate, then you'll go through that route. If you don't have that differential in the reducing power, then you'll go through the transamination route. Okay, so don't make too big of a deal out of that, I think. Just kind of recognize commonalities with the malate aspartate system that we already talked about. Okay, so once we have let's see, let me just... Okay, so we're either going to get malate, the oxaloacetate is going to be converted to malate in the mitochondria. Malate's going to go into the cytoplasm, and then the cytoplasmic malate dehydrogenase can regenerate oxaloacetate. Likewise, if we're generating aspartate from oxaloacetate in the mitochondria, that can go into the cytoplasm and be a substrate for the cytosolic aspartate aminotransferase and generate oxaloacetate in that fashion. Okay, so oxaloacetate in the cytosol then, that is the substrate for the key rate-limiting step in gluconeogenesis. So the phospholynyl pyruvate carboxykinase step, this is rate-limiting for all of gluconeogenesis. And the major way that it's regulated is through transcription of the phospholynyl pyruvate carboxykinase gene. Okay, so transcriptional regulation and modulating the level of expression of the enzyme is how the major way that gluconeogenesis is regulated. As far as the enzymatic activity, in some respects, it's the reverse of the pyruvate carboxykinase reaction in that we're removing one of the carbons as carbon dioxide. So we have a 4-carbon oxaloacetate, and we are left with the 3-carbon phospholynyl pyruvate. This is an energy requiring step, and the energy to make the phospholynyl pyruvate is coming from GTP. Okay, so we got a high energy phosphate bond from GTP that goes to a high energy phosphate bond in the phospholynyl pyruvate. Okay, so this is the key regulatory step for gluconeogenesis. So I'd like to just spend a moment and talk about the transcriptional regulation of the phospholynyl pyruvate carboxykinase gene. Now, shown here, the phospholynyl pyruvate carboxykinase gene, the promoter elements in the PEP-CK gene are similar to the promoter elements in the glucose 6-phosphatase gene. And the fructose 1,6-bisphosphatase gene. So you get kind of coordinated transcriptional regulation of those 3 critical steps. Okay, I'll just show it in the context of the PEP-CK gene though, because that's the rate limiting step. Okay, so in the promoter of the PEP-CK gene, you have the minimal elements, the Tata box, and all the just basic stuff. But the major regulation is occurring through elements that are sensitive to hormones. So you have an insulin response element. This acts as a negative regulator. A glucocorticoid response element that acts as a positive regulator in the presence of cortisol. And then a cyclic AMP response element, which is sensitive to glucagon signaling and protein kinase A activation and that axis. So just to cartoon this out a little bit, cortisol is a hormone. It's a steroid hormone, so derived from cholesterol, and it's made in conditions of stress, essentially. The steroid hormones, they all act through binding to receptors that act as transcription factors. Okay, so normally we think of a receptor being at the plasma membrane of a cell on the at the interface of the cell and its external environment, and you bind glucagon, for example, and you transduce a signal to inside the cell. With these steroid hormones, they function a little bit different. The hormone itself actually diffuses across the plasma membrane. It's lipid soluble enough that it can do that. And then once it's inside the cell, it binds to the glucocorticoid receptor. And the glucocorticoid receptor, once it's bound to the cortisol, it acts as a transcription factor. So it actually binds directly to DNA in these glucocorticoid response element sequences. That then helps to recruit the RNA polymerase II, and you get expression of the PEP-CK gene and translation into the protein, and then increased gluconeogenesis. So cortisol is one input into the PEP-CK expression. Glucagon is another. So glucagon, the G protein coupled receptor, is coupled to adenylate cyclase, which makes cyclic AMP. Cyclic AMP then activates protein kinase A. Protein kinase A then phosphorylates the cyclic AMP response element binding protein, CREB. And when CREB is phosphorylated, then it binds to the CREB response element and again helps to recruit RNA polymerase and transactivate the gene. Okay, so cortisol, this is the endogenous hormone that activates the glucocorticoid receptor. But this is a pharmacological target for anti-inflammatory reasons generally. But there are different drugs, prednisone, prednisolone, dexamethasone, probably others that I can't think of. But this is an important drug target, so you have to consider glycemia when you're giving these drugs. Okay, so gluconeogenesis is a fasted state process, and we want to turn it off in fed state metabolism. All right, and the way that we do that is, as you could imagine, with insulin signaling. So insulin signaling, that feeds into the AKT, or also known as protein kinase B. The protein kinase B then activates protein phosphatase 1, and recall from our discussion of glycogen metabolism that protein phosphatase 1 is like a protein kinase A eraser. Okay, so all of the phosphorylations on the cyclic AMP response element binding protein that were put on by protein kinase A, those are going to be erased by protein phosphatase 1 with insulin signaling. The AKT / protein kinase B, this is also going to be phosphorylating the FOXO1 transcription factor. That keeps the FOXO1 out of the nucleus, and you don't get transactivation from these insulin response elements. Okay, so insulin is acting as a negative regulator transcriptionally, and cortisol and glucagon are acting as positive regulators. That transcriptional regulation of the PEP-CK and also the glucose 6-phosphatase and fructose 1,6 bisphosphatase, that's a drug target for the treatment of metabolic syndrome and some types of diabetes. Diabetes, it's frequently kind of considered a disorder of how we handle dietary carbohydrates, and that's certainly true. But just as much, diabetes is a condition of inappropriate hepatic glucose output. Okay, so with diabetes, you have a problem with dealing with the carbohydrates that you put into your mouth in a meal, but you also have this inappropriate elevated gluconeogenesis happening, and those both can contribute to hyperglycemia that's characteristic of diabetes. Okay, so there's a TORC2 protein, and this acts as one of the alphabet soup of all this big blob of proteins that coalesce at the promoter of a gene and recruit the RNA polymerase II complex and promote transcription. So there's dozens and dozens of them. TORC2 is one of them. This makes a complex with CREB when CREB is activated by protein kinase A and helps to transactivate the PEP-CK gene. This PGC1 alpha, this is the PPAR gamma coactivator receptor 1 alpha receptor, I think. And this is another little transcription factor that gets activated, and then that helps to transcribe a lot of other genes and remodel the fasted state metabolism. Okay, the bottom line here is that this system can be pharmacologically manipulated with drugs that act as AMP mimetics, they're often referred to as. So remember AMP kinase? We introduced that, right? Did Dr. Holy or anyone else talk about AMP kinase? It's like a major intracellular energy sensor and modeler of intracellular energy. So when AMP kinase, when that has AMP around, that gets activated, and we phosphorylate TORC2, and then TORC2 doesn't transactivate with CREB, and we don't get gluconeogenesis, okay? Because gluconeogenesis is an energy-intensive process, and if we don't have energy in the cell, we don't want to be making glucose, we want to be burning glucose for energy. Okay, so normally AMPK phosphorylates TORC2 and puts it in the holds it in the cytoplasm in low energy conditions. The drug metformin acts as activating the same pathways that AMP would. Okay, so metformin activates AMP kinase, and that then helps to eliminate this transactivation of the PEP-CK gene and can help to treat diabetes by blocking hepatic gluconeogenesis. Okay, does that make sense? Okay, metformin is a complicated drug, so it's if you read reviews on it, there are many mechanisms of action. Okay, so this cartoon is showing the regulation of the phosphoenolpyruvate carboxykinase and the glucose-6-phosphatase gene and the fructose 1,6-bisphosphatase gene. They're all transcriptionally regulated in a similar way. And then this PGC1 alpha, that's that little kind of extra transcription factor that helps to amplify the sequence. I think rather than walk through this all again, I'm just going to leave this for your review. So essentially we've got glucagon signaling over here and insulin signaling over here. So take a look at that, and if you have questions about it, I'm happy to review it. Are there questions? Okay, I'm going to just introduce one concept, and I think you've maybe heard me mention futile cycling once before or twice. Okay, so futile cycling, this is referring to having a catabolic and a corresponding anabolic pathway active at the same time. So let's consider if you had gluconeogenesis happening at the same time you had glycolysis happening. You have phosphoenolpyruvate. This would go through glycolysis down to pyruvate. Then it would make oxaloacetate. Then it would go out of the mitochondria and be a substrate for PEP-CK. So if we went through this cycle, we wouldn't be making anything. We'd have a molecule of phosphoenolpyruvate here that would be the same molecule of phosphoenolpyruvate, right? But we would be hydrolyzing high-energy phosphate bonds. Right, and because this ATP, that's got to come from someplace, right? So we've got an ATP here and a GTP there. We're only making one ATP here. So every time we did this trip around, we would be hydrolyzing an ATP, and we wouldn't have made anything, right? So that's what we're trying to avoid. And what we'll talk about at the beginning of the next hour are strategies to turn off glycolysis when gluconeogenesis is on, and vice versa. Okay, so why don't we take a break and come back at 2 o'clock? That seems so late.
I'm used to it being like early.
You guys are good. All right, take care. If you do like the heartburn, however you say hi. You got
I'm home. I didn't. I played basketball, yeah.
Wow. That's not a question. There's a
Yes,
player, yeah. Not ready for it. I kind of like wrapped down the
Played basketball player weird. It was packed.
Really?
It was super weird.
That's weird.
We got really desperate Saturday. There was like it's like that. Yeah.
Did you like get in and then leave?
You know, got split off.
Good show. It's fun.
Yeah, nobody got black.